Unmute. Okay. Hello, Karen.
Hello.
You have been really active. That's nice. I've been reading your comments and your posts. Good job.
Thank you, Professor.
Nisreen, I can't say that about you. You should try to accumulate 2 to 3 to 4 points a week by doing activities. Do you have access to the Blackboard, Nisreen? Yes. Oh, I see. Okay. All right. So make an effort that you get to do something. Okay. We're going to wait for everybody to join us in 3 more minutes. So Karen, what was your impression of quiz 1 on chapter 1? How was it?
So quiz 1 was pretty good. There was one question that I think I got wrong. I can't remember which one it was. It was the one where you could pick more than one option. Yeah, I picked 3. It was probably 2, and I think I know which one I got wrong.
Yeah, I unless I look it up, I don't remember.
I don't remember. I took it like a week and a half ago, so I don't remember which one it was. But I'm a little disappointed that I got it wrong because I read the textbook. So it's okay.
So do you think, in your opinion, was it fairly fair in terms of what it covered if you only read the chapter?
Yeah, I think it was fair. I read the chapter, so I thought it was good. I reviewed the lecture, and I did pretty good.
Okay, very good.
It's fair. Thank you.
Morning, Professor.
Okay. Hi, Justin. Hi, Nick. Hi, Leroy. How are you guys doing?
Hello. Good.
Doing good.
Good. Let's see. I am trying to have a look at what you guys have done. I see Justin. Okay.
Yeah, I did just I still have like a few more activities to do. I know it's optional, but I'm looking forward to do it. So
yes, remember you can accumulate up to 4 points a week. So if you do beyond that, then it's just going to be for you to learn, but you're not going to be
I'm
credited.
on the process. It's taking it step by step. And also,
okay.
I still don't have
The textbook
someone to work with. I looked out those few names you gave me last week. So I don't know if they're here or not, but.
Okay. Well, we'll talk probably not today, because today we have to really spend time on doing what we have to do. But we will get back to groups, pairs on Thursday. Okay. So let's see, Nick. I think you have done pretty well, right? Let me see. Nick. Oh, yes. Fantastic. Good job, Nick, both on your blog entries and on your quiz 1. Leroy, I haven't looked up the grades for quiz 1 after whatever 7 p.m. yesterday, so I don't see your grade, but I see that you had 4 really nice blog entries. Good job. Good job. Okay. All right, everybody. Those of you who are here, remember, for you to get points, so to speak, for this class, which is 1, you need to do another blog entry, 200 words or a little bit more than that, either a summary of the lecture or a particular topic, or what would be more interesting questions if you have that other people could comment or read or do things like that. Okay, remember that this lecture is being recorded, so you will be able to get back to that if you need to. So without further ado, let's start doing that. Okay, share, and let's go in the sharing's paused. Why? Come on. Okay, guys, do you see the slides?
No.
You don't. Okay, so let me stop and do it again. Share screen. Share screen. Share. Okay, how about now?
No.
Yes.
Okay. Yes or no? Let's see. Karen Nisrian, do you see the slide called Chapter 2, Cognitive Neuroscience?
I see it.
Okay, good, good. So who doesn't? Okay, so everybody is in good shape. Fantastic. All right, so we'll see. It may not be full 2 hours, but what I would like to do is actually have a little break in between of two sections of the chapter, and perhaps give you a chance to, ask. Well, let's do it this way. If you have a question during the presentation, all you have to do is raise your hand or say something, write it in the chat, and then I will address this question. If you have other questions that are sort of general about the class and maybe finding five, partners, or whatever else you may want, let's keep it until that break, which I hope will be around 11:05, and I can answer your questions then. Okay, so anything that requires clarification immediately, do not hesitate to stop me and say I don't understand or slow down or whatever. But keep the general questions for the break. All right. Come on, let's move on. Okay, so let's see. These are a few. So Noor is saying I did not do it introduction. Okay, good. Noor, let's wait until the break, and then maybe we can start with you after the break. All right. So today's stuff is not particularly difficult or particularly new. It's covered in any introductory psychology textbook. So the only sort of twist that we want to have here is focus more on cognitive material. So let's go back and revisit the story of a 10% of the brain myth. Quite a few people in this class actually did the activity on that, which is nice. Then I would like to just go over a little bit about the hierarchy of the nervous system, in particular neuron, glia cells, and neuron structure. Just a couple of words about communication synapses and transmitters. All of that, like I say, is covered both in Intro to Psych, maybe not so good, but also in anybody who's taking bio classes would know that pretty well. So I'm just reminding you that what you need to know about this. Maybe mention the divisions of the nervous system. The one that we're interested in is the central nervous system, of course. We're not so much interested in the peripheral nervous system. It is important, maybe not as important for cognition as it is for other, let's say, motivation perhaps and personality. Most of the stuff that we really want to talk about has to do with the brain, the parts of the brain, a localization of function, stuff like that. And we will finish with the methods to study cognition. But textbook, the chapter in the textbook is designed in a sort of interlocked way that it starts with a couple of clinical cases and then goes into the neuron structure and the brain structure and then comes back to the method. So I'll try to keep that in mind. But again, you know, what's the point if I were to just tell you what's in the textbook? And hey, we don't have to be synchronous and you can just read it and be happy. But again, certain things are important. And if you feel like you are, whatever we're doing in chapter 2 normally would be known as biological bases. If you feel like you have gaps and it sounds all Greek to you, then like I say, go to anything like a textbook in Intro to Psychology. Here is one that a lot of our Psych 100 courses use, Psychology by Lilienfeld and colleagues. We have this unified approach that is most of our Psych 100 sections use this textbook. Okay, good. So again, remember, ask me a question in the process if you feel something is not clear or you are confused. Okay, so there are 3 main approaches that I will mention now. We will only spend time on approach 3 today. Let me get something. So the one that we are going to talk about today is this one, but we will be seeing the first 2 all of the time. So the first one is the sort of a core hallmark of cognitive psychology. That's mental representations. The problem with cognitive psychology is that we can't really see what we are studying. I mean, what's the mind? Yeah, you can have a surgery and you can see the person's brain, but the mind is not the brain. The mind is the result of the brain's function. So it's something that we infer from the physiology or from human behavior, which is what psychology is all about. So we'll be talking a lot about mental representations, and they will be present at every single level of cognitive hierarchy that we will be studying. We're going to be studying and starting with the lowest one, which is sensation and perception. That's the topic of next week. And we will go up. We will go to attention, and then memory that sort of penetrates the entire cognitive system. And then we'll look at things that are supported by these lower level of cognition, such as language and categorization and problem solving and all these other things. At every level, we will have a mental representation. So for, let's say, perception, we will have something that we will call a percept. Percept is our mental representation of the sensory experience. So if I'm looking at, and I'm looking at the tree now behind my window, I have a mental representation of that tree that has special characteristics. And it is mental, that's not physical. So it's different from the actual tree. And we'll talk about distal and proximal stimulus next week. That actually had to do with the question about the tree falling in the forest and nobody is listening. What will happen? So mental representations at every level will be something that we conjure as psychologists because we can see the reflex of that mental representation in humans' cognitive behavior. So at the memory level, we will talk about memory trace or use Carl Lashley's terminology engram. So that's point number 1. And we will be constantly referring to that, like I said, because the problem with cognitive psychology, I don't think it's a problem. It's just what it is, that we have to infer these mental representations. And the second one that you will be seeing in the textbook, and I will be talking about, especially historical approach to the topics that are coming our way, attention and memory and language, is what we call information processing. I already mentioned that the first lecture, but when I say information processing, I mean something like this. That is boxes and arrows. That is, these boxes are modules, right? So they are stores or buffers or steps or phases, whichever way you want to call them. And they are connected with arrows. So what happens at stage 1 then gets transferred somehow to stage 2 and then stage 3. So information processing approach is really boxes and arrows in a way. Nowadays, when we talk more about neuroscience, and that's what this class is all about today, we perhaps are less attached to this model of representation, but it's important, an important historical development that characterizes cognitive psychology. And finally, we will be talking about it again. That's why I'm saying these are 3 main approaches, because we will be mentioning each 3 of them at every class, in every class. So the third one is mind and body. That is, we know that the brain does the mind. Okay, so those psychologists who were called functionalists in the past assumed that to understand the mind, we need to actually study the nervous system and other systems that are related to it, because this will tell us something about the workings of the mind. That's why they're called functionalists, because it is about function. Okay, great. Now, going back to our little favorite theme about 10% of the brain, one of the questions that I asked you guys a week ago, actually, Thursday, was to perhaps, if you're interested, figure out whether this myth, and now we know it's a myth, came from. And so I've done my homework and what I found in the days when I was interested in doing this is that there was a book from 1956 by this person, Germain, called The Magic Power of Your Mind. Obviously, you want to unlock the magic power of your mind, so you have to say that some of your mind is not being used. So he says, the hidden 9/10 of your mental strength lies buried. Discover, release, and use it to gain new success and personal happiness, to full, richer life. And then the rest of the quotations actually come from all kinds of ads. And one of them is from the airline magazine in those days when we had in-flight magazines. But again, as you can see, all of that is, besides the book, all of that is late 1990s. Obviously, as cognitive neuroscience developed, it was easier to criticize this approach. Okay, so, hey, it is an amazing structure, right? Our brain. I think the philosophical conundrum is that how do we study something that's bigger than us? It's actually the most complex entity in the universe. It's not the galaxies, it's not the space, it's not the computer, it's not artificial intelligence, it's our brain. So in the sort of early estimates, it was suggested that the brain has about 100 billion neurons. Now, there is a new estimate that's actually a range between 86 and 100 billion neurons. As an interesting question for you guys, if you have time and desire to do so, is to actually find out how one could count neurons. I mean, if there were even a few 100,000, maybe you could actually do that. But how do you count 86 billion neurons? But in addition to these 86 or 100 billion neurons, we also have about 100,000 miles of axons. So we have really, really long axons in our body. These neurons connecting with each other give us about 1 trillion synapses. I mean, I can't even imagine this number. I mean, how do you even write it using scientific notation still? The blood that's being brought to neurons comes from 400 miles of capillaries. I mean, these numbers are just beyond anybody's comprehension. And there's 1 trillion glia cells, which are a different type of cells in the nervous system. And I'll talk about it in a second. All of that, that's amazing, is packed in this 3-pound structure, right? As you know, and we'll come to that at the end of the lecture, what is at the top of the brain is what we call the neocortex. And neocortex is about 4 millimeters thick. Now think of a credit card, which is a 1 millimeter. And this is just a bit more, 4 millimeters, right? And if we kind of take it out of this cranial form and spread it with all sulci and gyri spread, we're going to get a sheet about 20 inch by 20 inch, like the size of a big pizza. I mean, how does this happen? That's so amazing. So if one were to say that 90% of our brain is not being used, that is, hey, we could remove it, then we're going to get from 3 pounds to 0.3 pound, right? That's the brain of the sheep. That would be amazing if we could function the way we function at the level of a brain sheep. Of course, there is a correlation between the species size and the size of the brain. So an elephant has a much bigger brain than we do. But this correlation is not perfect. We have species that are smaller and have larger brain and vice versa. If you're interested, have a look at the ontology of brain development in any Wikipedia article. Anyway, so it's pretty amazing, but we're not talking about anatomy here, really. We really want to know how these organs, tissues, systems related to our human behavior. Okay, so what we really want to know is what are these psychological functions behind these different brain structures. All right. And again, there's so much stuff on the internet that I don't even want to bother to list things. You know where to find them if you are interested in sort of more biological approach. Let's start with a few clinical cases. In particular, there is one that's described in the textbook, but a couple of others that are mentioned in the textbook. Now, and one of the reasons why we actually could trace the origin of this myth of only using 10% of the brain back to the 19th century. So maybe some of you have seen that. This is called a phrenology head. Oops, let me move away this thing. I actually have it in my office, but of course I'm not in my office. So I can't show it to you because it was extremely popular. Now we know these days, we call it a pseudoscience. The idea is actually great. It's the implementation of this idea that's not really scientific. Now, it came to us from the work of two German, physiologists, Franz Josef Gall and his pupil and colleague, Johann Spurzheim. And they are known now in the history of psychology as the fathers of phrenology. Now, when you guys go to PSY288, which is History and Systems, you may be able to learn more about these. Were very interesting people. They're worth looking at. Now, so for them, the main idea was that the brain functions are localized. That is, now if you're talking about language, it's going to be in one area of the brain. If you talk about perception, I'm using modern terms. In those days, of course, they didn't. That's not to say that there were not other people who thought that we need to have a holistic approach to studying the brain. And Marie-Jeanne Pierre Florence is one of these people. He was a French, again, not a psychologist, but physiologist who thought of the brain as sort of a holistic structure. So this debate went on for quite a while. And in a way, it never ended. We continue to sort of think how we should approach biological bases. More recently, like 100 years later, Carl Lashley went into his research program, which had to do with looking for the memory trace. And he called that particular mental representation the engram. And he did that with rats. And in those days, ethical considerations were less of an issue than they are today. He would take his rats and he would train them to run a maze. That's what rats do the best. And he would then ablate parts of the brain. Ablate means he would create artificial lesions in the rat's brains. And he was looking to see at which point the rat won't remember the maze. And no matter how much stuff, of course, to a certain degree, about 80% of the brain was removed, the rats, not so fast, not so good, but actually most of them remembered the maze. So Lashley had to say, I give up. The actual engram doesn't exist by itself. It's sort of distributed across the brain. So in that sense, Lashley's approach was more close to Florence than to Gall and Spurzheim. So what was the phrenology? And because it was all the rage in the late 1800s, 1880s and above. So the main idea is that different parts of the brain, right, are connected to different psychological functions. So imagine that you meet somebody who is extremely obese. And in those days, if you are interested in phrenology, you would let a specialist trained in phrenology to actually touch your skull, go through. And imagine that somebody who's interested in this approach would touch the skull of an obese person and find a particular bump on that person's head. Hey, and lo and behold, going to the next person who is also obese and interested to find out what's going on with her or him would find almost similar bump. So it was a really easy way in those days to say, aha, this is where your sort of appetite lies in that area of the brain. Okay, so the main idea is the brain is composed of discrete organs. That's the phrenology idea. Each of them is associated with a different psychological function. Here is one of the 19th century charts. Now I want you to pay attention to, let's say, here's one, parental love, which means that's the love that parents feel toward their children. So you could imagine going to your parents and saying, well, let me try the occipital lobe of your brain and see whether you have a bump there. I mean, do you really love me as your child? Now, we of course know now from all the work in cognitive neuroscience that the occipital lobe is really about vision. It's not so much about parental love, unless, you know, it's in the eye of the beholder. A couple of other things that I would like you to sort of pay attention to is the benevolence and veneration. That's an interesting couples of areas, and interesting for a reason that will become clear in a minute. So the phrenologists, and of course Gall and Spurzheim, the main ones, but there were many others, came up with about a list of 40 or so of such faculties. Now, if you're interested about, let's say, some of you maybe read about spousal infidelity for week 0 that I posted, there is actually an area that phrenologists postulated for spousal fidelity. So you can check your own skull to see if you have anything special about it. And there is an activity that I ask you to do for this week, which will be open until next Tuesday, that have to do with phrenological faculties. So after today's lecture, you can go. I know that a couple of you have already done a couple of activities from this week. Now, so a very interesting case that is mentioned in the textbook is the case of Phineas Gage. Again, very classical case described in all kinds of intro to psychology textbooks. So very simply, this person was only 25 years old, and he was working in the crew on construction of railroads in the United States. It was very disturbing, Justin says. Yes. And he was the foreman of his team. And so they would put those pockets of gunpowder to blow parts of the rock to make tunnels. And of course, when they put the gunpowder into these holes, they temper them. So they pack them. So he was tempering one of those gunpowder holes, and there was an explosion because there was probably a spark. And what happened to the tempering rod? It went through his head in a very special way. I'm not showing you pictures, but it's very easy for you to find. It went from underneath the left eye, through behind the eye, and came out at the top of the head. And the reason why I showed you this, let me go back to, this, to benevolence and veneration, because that's where the tempering rod came out. So there was a surgeon not far from the place where Gage and his team were working. So he came very quickly. I found him conscious, which is amazing, of course. Then remember, that's an interesting point. Perhaps not everybody knows that the brain itself does not feel pain. That's just because our pain receptors are in other areas. It's the actual impact, of course, was painful, but he was conscious and he survived, which is the most amazing thing, especially in those days. But remember, he was only 25 years old. So he lived for another 12 years. And he traveled with a circus, and he would show his mutilated face. Of course, the eye, the left eye, was closed after the wound healed. And he had this tempering rod that he would show to people, and he would collect money like this. Now, but the most important thing about this whole part, right? So the damage at the top of the head was at the benevolence and veneration areas, right, that I just showed you on the phrenology head. And what happened to him is his personality has changed dramatically. Dr. Harlow, who was the surgeon who treated him, said that he was no longer Gage. So before the accident, he was good-natured, egregious guy. And after the accident, he became grumpy. He was cursing nonstop. He could not stop himself from impulses. So the idea was perhaps benevolence and veneration areas were damaged in him. So if you're interested, there is an interesting book by Malcolm Macmillan, I think his last name is, on Phineas Gage. And there are many other people who looked at his case just to see if they can be reinterpreted for the current thinking. That's not the only one, obviously. There is plenty of other interesting cases. In fact, one could structure this entire course as a case approach. So perhaps another most famous case is patient Tan. Again, a couple of you have already read a short article by Domansky. I think it's activity 2.6 or something like that in your Blackboard, where Paul Broca's, who was a surgeon also and a physician, observed this person whose name was Victor Le Borgne for about 30 years. And the problem with Victor was that he could only say one thing, and that thing was Tan. So he could say Tan, 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 nothing else. That's why he's known in the history of psychology as patient Tan. The only way to find out what happened to Tan's brain was to wait until he dies, which of course happened. And then Broca's did the dissection of his brain, and he found this particular area colored in green having a large lesion and the tissue was sort of shrunk around it. So Broca's made a case that this area is responsible for language. I mean, it's a little crude, but well, technically correct. And this area in the brain is now known as Broca's area. So here's an interesting question for you guys. Again, if somebody wants to do that, is to see, this is the left hemisphere, right? What do you think? Well, maybe you can answer it now in the chat if you want. Do we have Broca's area in the right hemisphere? All right. Think about it and type it in the chat at some point. We'll come back to aphasia. Noor says yes. We'll come back to aphasia. This is what happens when you have damage to one of the two brain areas. There's a second one that's right here called Wernicke's. And when we study language, which is going to be our chapter 9. And obviously, we will be talking quite a lot about patient HM. Of course, he died 11 years ago, so now we can use his name. And his name is Henry Molaison. And Henry Molaison is patient HM whose hippocampal structures were removed during a sort of experimental surgery in 1956 or 57, after which he became profoundly amnesic. So we know that hippocampal structures are implicated in laying new memories. Again, these are important cases, and we'll come back to them as one of the methods that cognitive psychology uses to study cognition. Okay, at this point, we can move on to the organization of the nervous system. In this case, we would want to talk about the neurons and the actual central nervous system, which we will be, well, we'll talk quickly about the brainstem, but mostly we'll talk about the actual brain. So let's start with the neuron. But before we do that, I see a couple of people in the chat have a little conversation about whether we have Broca's area in the right hemisphere. But I'm going to ask you now to type an answer to this fill in blank space. And I can see there is 14 of you guys, so it would be really nice if everybody typed in something. So I'm asking you to fill in the blank in the sentence. All cognition is the result of what kind of activity? I'm going to give you a minute to do so. Okay. A bunch of people say brain, but the correct answer is, who is getting the mental activity? All right. Roy. Isaac. Isaac gets the prize. Neuronal activity, that is the activity of neurons. All right. Well, we can say neurological, but it's just a synonym, right? It's our activity is the result of, yes, Isaac. Go, Isaac. Good. Is the result of neuronal activity or neurological activity. Of course, that's electrochemical, but that's sort of the next step. That's how neurons work. Okay, good. Trying to scroll down to the end of the chat. Okay, so that obviously we have to predicate all our cognitive behavior, that is thinking, language, categorization, and all of that as a result of this neuronal, neurological activity, whichever way you want to do that. So brain, of course, is as a layman's synonym is fine. It is neurons. But we have to say more than the brain, because neurons obviously are not just in the brain. So let's talk about the organization of the nervous system, right? So at the lowest level, we have cells. Going one level up, we will have tissues. That is the groups of cells. And I'm intentionally being vague for a minute. See if you can perhaps, you don't have to type that, but think. Can you give a definition what a cell is? Can you give a definition what a tissue is? It especially should be doable for those of you who are taking biology classes or who are thinking about physical therapy or any other sort of physiology related education later on. Now, of course, groups of tissues will give us organs, right? And organs will be grouping together to give us systems. And finally, we're going to get to the organism. That's the highest, right, hierarchy for us. And that's a living individual, a human being or an animal that composed of many separate but interrelated or interacting systems. Okay, and of course you guys are familiar with this classification of from species to kingdom. Again, not a part for our class, but something that you study in biology. Now, if you thought a little bit about what these issues are, what these terms mean, let's look at the actual definition. So the cell is the smallest unit that can function independently, right? So anything within the cell is not going to be functioning independently. So it's not going to be the smallest unit. So tissues are now groups of cells, right? And they have to be of a particular kind. Now, organs, groups of tissues that perform a particular function. Now we have groups of organs that become a system, and in the system they perform related functions, and finally we have this organism. Okay, so now let's take this classification and apply it to the central nervous system. That is what is important for cognition. Okay. Come on, thingy, move. There we go. All right. So for the nervous system, these are the two types of cells, so two basic types of cells that we're interested in, neurons and glial cells. Now, we are sort of used to thinking about neurons as the most important type of cell in the nervous system, but glial cells actually are, especially recently, there's been some research that shows that glial cells also perform particular important for cognition functions. So these two. Now the next one are tissues. And in the nervous system, these tissues are nerves, ganglia, and nuclei. Again, we probably won't go into these details. It's just for you to think about. If you take another system, let's say endocrine system, what would be the cells and the tissues in the endocrine system? Some of them will be different for the nervous system. Organs are, of course, the three that we will talk about soon. And these are going to be brain, brainstem, and spinal cord. And spinal cord and brain are connected by the brainstem. And finally, we have the two components of the nervous system. One, the central nervous system. Again, that's what we are interested in. Then there's the peripheral nervous system. Important, maybe not so important for the purposes of cognitive stuff, but important for other things. No. Come on. Why don't we want to move? Here we go. All right. So again, I expect that you know all of this, especially not only because some of you are already far advanced in your studies, many of you are thinking about professional degrees, and you all have taken not only Psych 100 but bio courses, just in case you are rusty a little bit on that. So let's remind ourselves what neuron is and what its structure is. Okay, so it's a basic building block of the nervous system. Remember, cells are actually at the bottom and the nervous system is at the top of the hierarchy. So we have here, we have a cell body, right? That's this pink stuff. It has a nucleus right here. Obviously, nucleus is smaller than the cell, so it's not an independently functioning unit. Then we have those little things called dendrites, right, that are here. And then we have the very long thing called an axon that connects this neuron to the synapse, right? And at the end of the axon, we have those sometimes called terminals, sometimes called buttons, that release neurotransmitters, whatever. Okay? So each of these elements has its own function, as you probably, again, are familiar with all of this. So these dendrites of a neuron receive a stimulation from the adjacent neuron through the synaptic cleft. These are, in the sense, afferent, AFF, portion of neuron. That is the neuron. They receive the sensation, the feeling, so to speak, from the other one. So if you, it's not actually the sort of ending branches that are responsible. Okay, before I continue, my internet tells me that it's unstable. Can you guys see and hear me? Just type yes. Uh-huh. Okay, good, good. Everybody is good. Okay, perfect. So whatever my internet says. So here's a question about axon hillock. Axon hillock is actually the same as axon terminal or axon buttons. It's just a variety of terminology in the field. So hillock, I think, is more of a older term than terminals or buttons. So you can use them interchangeably. Okay, so when the neuron is sufficiently stimulated, right, what happens? The cell body discharges an electric impulse, giving us the neuronal communication. This impulse is carried through the axon to these terminal buttons or axon terminals or hillocks, all of these, right? And this is going to be the efferent portion of the neuron. It stimulates other neurons. So if you need a mnemonic, think about afferent is receiving the stimulus. It's here, right? And efferent is about effect, about doing something. And that's the axon terminals at the end. Now, what else we have here? Again, this is all very easy stuff that you can read anywhere else. We have the myelin sheath. It's this sort of fatty white stuff that carries the neural impulse, and that plays an important role in the damaged peripheral nervous system. Anybody who knows about MS, multiple sclerosis, knows that myelin sheath is implicated in MS. And this is, if you look at the brain when the brain is open, I've seen that quite a lot because I work at the center where there is a open, awake brain surgery for people with gliomas. You will see a lot of white stuff. When you see that white stuff, that's the myelin sheath. Not all neurons have myelinated axons. And there, again, there is a difference between those. And this stuff, let me go back to the cell body, actually is gray when you look at the brain. So when you see the white stuff, you see the axons, the white matter. When you see the gray stuff, you actually see the gray matter, which is the bodies of the cells themselves. Here on the axon, and the axons can be very long actually. We have some axons that are whatever, tens of inches long. These are called nodes of Ranvier, okay? And they are sort of interruptions in the myelin sheath. And the number of these interruptions of these nodes actually affects the speed of neural impulse. Okay, so again, there's absolutely nothing new in all of that. That's very well explained in all kinds of other textbook. But you know, if you're interested and if you are a kind of person who likes doing things with your hands, I have posted a little activity for you. You can color a neuron and label its components and their functions, and either scan or take a picture and post it on your blog. You know, it's kind of interesting way to emphasize things when you do them as opposed to just listening. All right, like I already mentioned, we have three types of neurons. We have what's called afferent with an A neurons, we have efferent with an E, and we have neurons that connect afferent and efferent neurons. So afferent are the ones that carry sort of the sensory information from the receptor organs to the spinal cord and the brain. So think about hearing, right? So there is a neuron, there are neurons that will connect through the very intricate inner ear through the spinal cord into thalamus, and from thalamus it's going to go to the temporal lobes and so on. And the efferent ones are the ones that go the other way around. They carry motor signals from the spinal cord and the brain to the muscles and require us to kind of do things. And the interneurons, obviously, judging by the name, are the connecting ones. Okay, so neurons is everybody knows what neurons are, but glia cells, that's what I would like to spend another minute on. Glia cells are actually much more numerous than neurons, but they also a type of elementary unit in our nervous system. As I said before, if there is 100 billion neurons, there is almost 1 trillion of glia cells. So 10 times as many. Okay. Maybe, I don't know, maybe that's why there was this idea that there is 10% of the brain that we use, because if we only use neurons, but there are glia cells that are 10 times as numerous as neurons, that could be the source of this idea. But we have to say that all of our neurons are engaged in mental activity. All right, so what do these glia cells do? Well, they build a myelin sheath around axons, some axons, not all of them, and that provides the insulation. So as you perhaps know from physics, electricity travels better through the cables that are insulated. They also help with migration of axons and dendrites during development. So children develop all these intricate connections in their nervous systems. This migration and interchanging situation helps neurons to be more efficient in connecting with each other. They also do to sort of keep neurons in place so that they can so they pack them in a way so that they can keep, well, they are in the proper places for synaptic transmissions, and they also help transfer nutrients and remove waste in when like neurons die. When they die, they have to be cleaned up. So that's what glia cells do. So for a while, like I said, it was believed that the glial cells don't really do much in terms of mental processes. But there is now some interesting evidence that they can be um, a part of information processing and communication network of sort of their own. They run parallel to neurons. And if you're interested, I can give you a URL to read about it. So there is a professor, Marian Diamond, who actually is famous for the neuroanatomy course. If you're interested, I was going to show you this book. Oops, let me do it now. Oops. Okay, so that's not hers, but it's a book on sort of coloring the various components of human brain, and I'll show you in a second, but I can show it to you now. Here is, for example, part of midbrain. I don't know if you guys can see that. If you want to really learn, if you're going to be a physical therapist, for example, doing coloring activities actually help a lot. Anyway, so you can have a look at Marian Diamond's work on functioning of the glia cell. So if we think about Einstein's brain, that's a famous story. In terms of its neuronal structures, it doesn't seem to be anything special. It's just another brain. However, what people who've been studying Einstein's brain discovered was that it's the glia cells that are different in his brain. That is, there's very high concentration of glia cells in Einstein's brain. So perhaps this is about metabolic activity between the neurons, and that gave him that edge. There's also some findings that his corpus callosum, which is the bundle of axons, the white matter that connects the left and the right hemisphere, is actually much denser. It's bigger than average, perhaps allowing for greater connectivity between the two hemispheres in his brain. Now, again, we know that he was famously a visual thinker. That is, he really wanted to see things. And that's why I'm saying, hey, use a coloring book if you would like to learn more about the nervous system. And again, there's an interesting article in Scientific American on, it's called, let me see my notes, Finding Einstein's Brain. Interesting, from 2018. Now, glia cells, and that's one of the reasons why I wanted to mention them more than usual, they seem to be implicated in certain forms of brain disease, in particular Alzheimer's. So, as you know, in Alzheimer's, a patient suffers dementia, which is a general loss of intellectual functions. It's usually more pronounced in elderly people, right? 80s and above. But sometimes it happens in people in the 50s and 60s. So it is, as we know now, is the result of a degeneration of nerve cells. Sometimes it's just natural. It progresses the way it is. Sometimes it's due to some, genetics, so it's accelerated. And in either case, the dead neural tissue is replaced with glia cells, okay? Resulting in what we call plaques and tangles that are so characteristics of Alzheimer's, and only discovered really at autopsy after the person's death. So if you're interested in looking at the really relatively new is the past 15 years, the research on how we can diagnose AD in living people without waiting until autopsy, I suggest you do this activity on the blackboard. And we'll keep talking about AD when we get to memory later on. So let's quickly finish about neuronal activity in synapses and have our 10-minute break. Okay, so here we have our body cell. that discharges an impulse that travels across the axon to the axon buttons, releases the neurotransmitters. And what we know about this neural impulse is that it's all or none. There is no volume modulation. You know, neuron cannot fire stronger or weaker. Neuron can only fire more frequent or less frequent. And, whoops, let's go to the next one is something that I hope you know. Neurons themselves physically are not connected, right? They don't touch each other in any meaningful way. They are separated by gaps that is now known as synapse. It was a term that was coined by Charles Sherrington, and we'll talk about him next week when we talk about perception. So those neurons are separated with this little space, and that space is really, really small. And so we call the neuron that's before it, the one that's sending the signal, as a presynaptic neuron. So sending neuron, that's this guy. And the one that follows, receives the signal, is called a postsynaptic neuron. Okay. So each neuron contains these axon terminals, axon buttons, and so could be connected with as many as another 1,000 neurons in its vicinity. The density depends on the type of the neuron and the area in the brain. Again, if you're interested, there is a lot of literature on that. So with 100 billion neurons plus 1,000 synaptic connections gives us approximately 100 trillion synapses. So we have more synapses than we have stars in our Milky Way galaxy. That's pretty amazing. And the last thing that we want to mention is the neurotransmitter. Again, there's a whole, if you're interested, not only the bio courses might help you, but taking PSY232, which is our biological psychology course, you will be spending a lot of time learning about the neurotransmitter mechanism and the kinds of neurotransmitters that we have in our body. Again, I don't want to spend more time than I have to because it's all well established in the literature and other textbooks. And I think at this point, let me check. Yes, let's take a 10-minute break. So at this point, we have 11:08. So let's reconvene at 11:18. I'm staying put. So anybody who wants to ask me a question unrelated to today's lecture or related to today's lecture can do so. Go take your technological break, go to the bathroom, have a drink, have a smoke, whatever you're doing, and come back in 10 minutes. Okay? So I'm gonna pause recording for a second. To the second part of today's lecture, the brain. Okay, let's go. All right, so this is our nervous system. It's a very simple representation for you to think about. Like I said before, we are not going to be talking about the peripheral nervous system just because we don't really have time for this, but the interesting point is, actually, I'm going to stop this presentation for a second and show you today's article in New York Times. So I'm going to share something else. Whiteboard. Where are we? Nah. Hold on a second. I have to make sure that I have it open. Come on. Why don't you want to? All right, I guess it's not gonna happen. Maybe later. Anyway, so there was a story if you can access eligible for what? Oh, okay, let me answer these two questions. So eligible for what is whether you're eligible to do honors thesis at the Department of Psychology. That is, you as Verrazano students, maybe some of you even Macaulay. Do we have any Macaulay among you guys? Well, all right. Even Verrazano students, that's our CSI honors program. You are required to have a capstone project, as far as I know. Is that correct? Okay, Leroy says yes. Okay, so if you want to do a capstone project, now, of course, you need to do it in the lab, and you're not going to be doing it as a literature review. So if you are eligible, that is, if your GPA in psychology is 3.5, then you can do an independent study that will result in an honors thesis. In fact, we require 2 independent studies back to back, which is normally carried out during your senior year. You create a topic, you run your experiment, presumably, and you write a thesis. Leroy, in terms of whether we'll be back on campus in the spring, as so far they tell us that the courses are all scheduled as hybrid, which means there will be potentially certain amount of classes, parts of classes that might take face-to-face. Students can take face-to-face. But there will be also fully online synchronous courses, more like this one, and fully online asynchronous courses. So for us, for faculty, full-time faculty, I'm a full-time faculty, we have a choice. We can, at this point, we can opt out of hybrid and say, I want to teach online synchronously or even online asynchronously. In terms of opening the labs, that will definitely happen, but it's not going to be full functioning. It's not going to be the way it used to be. Come anytime, whenever you, it will be this step-by-step reopening, or I call it revamping of research. So yes, if for those of you who are graduating in June 2021, you would need to start now, and you can have an independent study still until September 15th, which means you need to approach the faculty that you're interested in and say, what can we do this semester that would not require on-campus work? So you could design your experiment, you can design materials, you can learn a platform like Finding Five so that you can run your experiment. If it's not an animal experiment, of course, human experiment, you can run it remotely. And hopefully in the spring, there will be a possibility of coming to campus, to the lab, certain time. Okay, so let's move on. Otherwise, we'll be sitting here for a while. So the central nervous system, the one on the left, is the one that we're interested in. And there are 3 major components. The spinal cord, which is basically neural impulses that are being moved between the periphery of the body and the brain and brainstem. Actually, they're kind of hard to separate. And there are a bunch of different tracts, bundles of nerves. Remember, nerves are the tissues in the hierarchy. That's why if an accident happens and the spinal cord is broken, it depends which of these tracts are severed. That will determine the kind of injury that will result. Again, we won't be talking about the spinal cord because we don't really have time for that. The terminology that we'll talk about in a second comes from the terminology of anatomy of animals. That's why it's hind, mid, and forebrain. The brain stem, like I said, is kind of difficult to separate from the brain and the spinal cord. It's in between. But anatomically, we separate it from everything else. And we say that there is a hindbrain and the midbrain. And like I said, this is because in most animals think that the spinal cord is parallel to the ground, right? It's horizontal. So you have the hindbrain at the back and the forebrain at the front. Now, of course, and the midbrain is in the middle. Of course, humans walk upright. So whatever is at the bottom is the hindbrain. Whatever is at the top is the forebrain. So that's kind of the way we think about it in humans as opposed to animals. Like I said, I would suggest that you guys do a little bit of coloring activities. In any coloring book would work. So here, different colors represent different areas. So spinal cord is this where it starts, right? We also have two other, so now this is the spinal cord. This is now the hindbrain. All of that is the hindbrain. So this is pons, and this is medulla. This is cerebellum. Cerebellum is a very interesting structure. Not that we have much time to talk about it, but if you're interested, I would suggest that you read about it. So medulla, that's the red stuff at the bottom, is responsible for vegetative functions like cardiovascular system, that's your heart and your blood, and things like breathing, so respiratory system. These are regulated by medulla. It's right here. And above that is the pons structure, and the pons structure regulates arousal, that is your sleeping and your awake cycles. Again, if you're interested, not that I have time to talk about it, damage to these structures, which is not that easy to get because it's really buried in your body, result in very interesting conditions. Perhaps you heard about the locked-in syndrome or persistent vegetative state. Depending on the damage to the structures, you can have coma, you can have either of these conditions. But it's extremely interesting because it's about the mind and brain kind of duality. So you think about the contingency table. So your brain is damaged, so to speak, one of the brain structures. What happens to your mind? And we used to think of people in persistent vegetative state that they are brain dead. But it turns out that's not necessarily true. We have cases well documented now where people appear to be brain dead, but in fact they're locked inside their bodies, and there's not much that they can do. There's a very famous case of a French journalist who had a stroke. Not an unusual thing. He was relatively young. He was in his 40s, where he was actually driving when it happened with his son. So when he came back to his senses in the hospital, it turns out everything was paralyzed in his body with the exception of one eye. So he could blink with that one eye. And once they discovered this, the doctors in the French hospital, I'm just giving you the gist. One of the journalists worked with him in the last year of his life. He managed for a few years. The journalist and Dominique, that's his name, Dominique managed to create a system where blinking could represent a particular letter of the alphabet. So Dominique wrote the entire book just through the blink of his eye with the help of this journalist. And there is a movie called The Butterfly and the Bell Dive, something like that. If somebody is interested, let me know. I'll look it up. I don't remember the exact name of the movie. It's very Justin, if you're saying that Phineas Gage is disturbing, wait until you read about Dominique Bobby. That's really disturbing. Okay. So what you could do, so cerebellum, like I said, means a cerebrum, like a small version of the brain. It has sulci and gyri, just like the regular brain. Usually what we know about cerebellum is about integration of sensory and motor data, especially in the balance for people. That's why perhaps if you've noticed that people who are severely plastered, that is severely drunk, cannot really walk well because they can't keep their balance. And that's because the cerebellum was not functioning at the moment. Okay, so the pons and medulla, I already said what they do, and I suggest a fun activity to you guys. If you are interested, when we talk about memory, we'll talk about, yes, of course, we have a lot of external ways to help our memories, but in the old days, when we didn't have, yes, Justin, thank you, the diving bell and the butterfly. Yay! So watch the movie, guys, if you have time. Thanks, Justin. So I was going to say that, I now lost my train of thought. No problem. All right. Oh, the cerebellum. I was continuing my story of the cerebellum. So it can be impaired by alcohol, like I said, especially in acute intoxication. If you just have a bottle of beer, you're going to be okay. But if you drink a lot, and if you drink a lot chronically, you will have this problem with your cerebellum. Your balance will be off. And now we actually think that cerebellum has more to do with cognition than we used to think about it. In fact, it has 3 times, it's one estimate, 3 times as many neurons as a cerebral cortex. Hey, so they're there for a reason, right? So if you're interested in cerebellum, do some search. And then coming back to the memory, I was saying that in the old days, when we didn't have our phones with us all the time, we had to rely on our memory. And to help us remember things, we came up with this device called mnemonics. And a mnemonic is just a technique that helps you to remember things. The idea behind a mnemonic is that you have to connect the stuff that you want to remember. Let's think in this case, you want to remember what the pons structure is responsible for, what the medulla is responsible for, cerebellum, and all of that. It's kind of difficult to try to just memorize it as is, but if you use a mnemonic, that will help you to memorize the stuff you could actually remember. So go to Blackboard, I think it's activity 2.3 or 2.4, something like that, that will take you through the mnemonic. And then one of them is called Pavlov's Really Freaking Mad. That's pons, reticular formation, and medulla. And there are pictures. So you associate the terms with the pictures, images, and that helps you to come back to what these structures are in a flash. You don't need to think twice. And the other one is Hatch, which we'll get back to when we get to the limbic structure in a few. Reticular formation is kind of hard to depict. I think I already showed you in this book. Now let's see if I can kind of point it out to you. Here's the reticular formation. It goes sort of inside the hindbrain, so it's kind of hard to depict. And reticular formation is about also cortical arousal. Anybody who has cats? Parker has a cat. Nisreen has a cat. All right, so you guys may want to cover your ears. There have been a lot of studies, anatomical studies with cats. So one could create a lesion that is damage the reticular formation on the cat. Obviously, remember, this is a hindbrain. That means hind and midbrain. That means these structures exist in all mammals. That's why these are evolutionary older parts of the brain. So cats have them and mice have them and so on. So if you remove that reticular formation from the cat will be either completely asleep forever, and you can only try to wake the cat with a loud noise or other intense stimulation. But if you leave it, don't remove it, but constantly stimulate it, which we can do with a single cell electrodes, right? The cats will be constantly awake, and they will be, crazily doing things nonstop until, of course, we stop doing that, and then they will return to the normal wake-asleep cycle. And the same with people. So coma often is a result of reticular formation damage, which is not necessarily mechanical, but more like strokes and stuff like that. Again, there's plenty of literature on that if you're interested. Okay. Right, so learn the mnemonics to memorize the hindbrain and the midbrain structures in activity 2.4 on the blackboard. And when we come back on Thursday, you can surprise me by telling me quickly what's in the hindbrain and the limbic system. Again, like I said, why we study these parts of the brain is because they are so interesting in terms of the kinds of conditions that result when there is damage to different parts of the brainstem. And I'm not really telling you which one does what. If you're interested, please read about them. And that's something that you could do. You could do that as an additional activity that you can post in your blog. Just don't forget to post the URL. Okay, our final piece is not final, but getting to the final one is the cerebral cortex and the limbic system. That's the parts of the forebrain, and the cerebral cortex is on the outside, so it's easy to, that's what we think when we talk about the brain. But the limbic system is buried within the brain. We can't see it unless I don't have my model of the brain here, which I do have in my office. So again, it's not something that's so difficult to find if you're interested to learn more about it. Okay, so remember I said that we have the brain weighs about 3 pounds, and the thickness of the neocortex is only 3 to 4 millimeters, but that's 80% of the brain, because that's where most of the high-level cognitive functions reside. And the limbic system obviously is something that is important for cognition as well, because it includes things like hippocampus, for example. All right, again, I suggest that you do some coloring. There's plenty of places to find. So as you know, we have the two hemispheres, the left and the right one. So whenever you see the cerebellum in the back, this is the cerebellum, all right? You know that this is the left hemisphere. Now, if it were the mirror image, it would have been the right hemisphere, right? So, and the way we think about the two hemispheres, what happened to my mouse? Wow, there we go. Where did you go? All right, when we look at the top of the brain, we will see what's called the cerebral commissure. That is the seam that connects the two hemispheres. And the two hemispheres I already mentioned before when I talked about Einstein's brain is connected with the corpus callosum, which is white tracts. These are the axons of neurons. And it's a very important structure because in the 1950s, that was one of the reasons, one of the structures that was experimented with for people with epilepsy. Now, epilepsy is a really, really difficult disease to fight. So one of the suggestions was that perhaps if the storm of epilepsy originates in the left, if you sever the corpus callosum, you can prevent the storm, this electrical activity, to spread into the right hemisphere. So there was a time for about 10 years, might be 8 years, when there were quite a few surgeries called the split brain surgeries, where people's corpus callosums were cut. Now, first they said, hey, it looks great. People seem to be intact. They seem to be normal after that. But it turns out that's not quite the case. There are sometimes subtle, sometimes not so subtle problems with the corpus callosum, not removal, but cutting. But for many people, it was a lifesaver in terms of epilepsy. So now, depending on those sulci and gyri that we have in our cortex, we distinguish the 4 lobes. Again, something that I'm sure you guys are familiar with. We have our frontal lobe in yellow here, which is the biggest one. We have a smaller green here, occipital lobe. We have the temporal one depicted in magenta, whatever it is, teal here. And we have the parietal lobe, probably the most mysterious one of all 4s. And here we have what's called a Sylvan fissure that separates the temporal lobe from the parietal lobe. It's well kind of, it's very nicely visible when you're looking at the brain, and so on. So there are fissures or gyri and sulci. Okay, the separation between occipital and parietal is not so clear, and it's interesting that for many people, some of these anatomical differences are very similar. Most of them actually are, but then there are certain ones that show individual differences. And again, at this point, we don't quite know why it is the case, but the science will continue to work with that. So the picture on the right shows the same sort of structure of the cortex and the forebrain, but cut sagittally like this. So you can see what's inside. So for example, the brown structure is the thalamus that we think of as a relay station for all kinds of sensory information, but you can't see it if you're looking at the brain at the cerebral cortex. You actually have to kind of cut it up like this. All right, and then the white stuff is the limbic system on the limbic lobe, whose structures are depicted here. Again, there's nothing new that I'm telling you here. All you have to do is read more about it in any other textbook. Interestingly, there is this green structure that is hypothalamus. So if you are hungry, hey, lunch is approaching, or thirsty, or if you want to have sex, all of these functions are regulated by the hypothalamus. And there is a wonderful little structure here called pineal gland, which is not a part of the nervous system, it's a part of the endocrine system. And that's the only structure in our brain that does not have a pair in the other hemisphere. It's only present in one. Okay, so I just named a few of these fishes. The original name, another way of saying fissure is sulci. That's plural, sulcus is singular. And we have this longitudinal, central, and lateral that separate the lobes. Like I said, the only lobe, the parietal versus occipital, is kind of hard. There's not really a fissure that is there, but we know a lot about the functioning of the occipital lobe because it's vision. Interesting point that I don't want to spend too much time on is to show you phylogenetically how our brain developed from a sort of evolutionary from a point of view of a species. So here's our friend, the rat. As you can see, there are not too many sulci or gyri here, but we do have a cerebellum, right? So we have the hindbrain here, all of that. Then comes the cat, and the cat looks pretty good, very close to what we are. Again, we now see some sulci, right? Here and here, for example. Again, a nicely developed cerebellum. And here is the human brain that you recognize. Again, the way our structures are laid out is a little different because we walk upright. There's also an autogenetic development of the brain. There are tons of really interesting clips on YouTube on how a fetus brain develops. If you have time, check it out. Write something about it in your blog. Again, remember, I would like you to be flexible. You don't have to do the activities that I propose. You can create your own activities, which would be, again, something for you to earn points for participation in class. All right, so finishing this part and moving to the last component, which is going to be the methods. Think about neocortex, and there's a lot of interest in the arts and the literature about the brain. So people who are artistically inclined and like doing photography, now there's a way of sort of doing something like this. So this is what you see here on the left is a photograph. Obviously it's colored to make sure that we can see that of the neocortex. Remember the 4 millimeter depth of the cerebral cortex, and you can see that there is there are darker stripes. Okay, I'm trying to sort of get one. And you can see that there are different layers. Although it's only 4 millimeters, it has layers, and these colors represent different different cells, different neurons that are found in the brain. Again, so there are a couple of interesting books. There is even a coffee table book that has just this beautiful blown up photos of different brains. So that is the book by Carl Schoonover, published in 2010. And he created a very nice sort of slideshow that you can access through this article, and that is posted as your 2.5 activity on the Blackboard, if you're interested in doing that. Okay, moving on to the final component of the forebrain is what is known sometimes subcortical structures, because they're underneath the cortex. You can't see them unless you open up the brain like this. Like I said, thalamus is known as the relay station. That's the sensory information gets to thalamus, and thalamus directs this information to various spots, various areas of the brain. So if visual information comes in, it directs the visual information to the occipital cortex, to the vision areas, of which we have quite a few different ones. If it is things like eating, drinking, and so on, that's a hypothalamus. Again, not really part of cognitive network for us that much. But there are two structures that are really important. One of them is hippocampus. That's the area just surrounding the thalamus, and we have two of them, like everything else, in the right and the left. And in HM's case, in Harry Melayson's case, not only hippocampi themselves were removed, but the area surrounding them, creating this incredibly dense amnesic case. And amygdala, I didn't point it out when I talked to you a few minutes ago, but again, it's easy. It's in the back, responsible for mostly emotions, anger, fear, and just being responsive. So there are several interesting cases that document uh, damage to amygdala where the patient becomes emotionally dumb, frozen. There's nothing that, I think it's one of the most famous one is a woman. So she was completely uh, uninhibited in her behavior without being emotionally involved. So again, read about that if you're interested in. We'll come back to hippocampus definitely because it's an important part of the memory stuff. Again, so here's your amygdala that I just mentioned, emotional center of the brain. And this is the hippocampus that looks like you know that the name means the seahorse, looks almost like a seahorse. And you can only see it within the, the brain because it's not visible on the surface. A final little component, I don't have much to say about it only because the textbook gives you a pretty good description of methods, but you could also look them up um, on the web and see if anybody is interested in real sort of brain dissection. You can let me know. I can direct you. And remember, coming back to the beginning of the lecture, that's the phrenology stuff. The idea was good, but the science was bad because we know why. It's not a scientific method. But the neuropsychological research that continues usually in practice. So those of you who perhaps are interested in clinical psychology or neuropsychology, to be precise, will be working with patients who have different conditions that affect their cognitive behavior. And then you have to find out what is that's wrong with them by using neuropsychological tests. And we know about neuropsychology because of the patients, because of these clinical cases. And they continue to inform our science. But to completely diagnose the person's condition, we have to wait until the person dies. And we do the autopsy. So it's post-mortem examination. Of course, with cognitive neuroscience in place, we are interested in doing it with living people. That's much more interesting and important. So what we are doing now, those modern faculties as opposed to phrenological faculties, is quite different from what Gall and Spertheim envisioned. But it's an interesting sort of segue to how cognitive neuroscience evolved, actually paying attention to what phrenologists had to offer. Again, just to remind you, the two important cases, Broca's patient ton, that's activity that you can do by reading a short article on the Blackboard. In the patterns of language disorders that we know as aphasia these days are quite different in Broca's and Wernicke's aphasia. I'm using sort of the classical terms. People who work with aphasic patients actually don't use it that much anymore. It's much more detailed. But if you have ever known somebody who had a stroke that affected either Broca's or Wernicke's area, you know exactly what I mean when we say the language is really impaired. And there is some rehabilitation that's possible, but it's often not so good. Often, especially when Broca's area is damaged, the person can't really talk much. It's laborious and slow. The Wernicke's people, the Wernicke's patients are very talkative. They're real chatterbox, but what they say doesn't make any sense. Again, Google that and look up the cases of sort of neuropsychology of language, known as aphasia. So do that activity if you're interested, or do something else that has to do with, let's say, Wernicke's aphasia, and post your entry in the blogs. Okay, moving to what we can do with the actual living people. We do have electrical stimulation. It's less so used with humans anymore. Occasionally, when there is an open brain surgery, if people, especially for epilepsy people undergoing certain surgery, the surgeons sometimes can insert electrodes, and it's kind of funny, right? Because you can imagine if you have something like this in your brain, but if the surgery needs to be corrected, sometimes people live with these electrodes in the hospitals, of course, for several days. So then you can imagine what it looks like. Let me see if I can show you a single cell recording, a historical clip, if it works. Give me a second. Control Alt Delete. No, if we can do that. Oh, all right, a second. And we'll be done in 5 minutes. I'm trying to get rid of the PowerPoint. There we go. Yeah, okay, good. Discard. Okay, where is the... Okay, let's see if that will work. Share the screen, media, share. It's a very short clip. Tell me if you can see it once. Can you guys see it? Yes, okay, good. All right. Good, yes, everybody says yes. Okay, good, good. So let's start from the beginning.
We can't hear it.
Ah, you can't hear it, I see. Okay, so let me, because it's short, it's only a minute. Let's do it all over again, and I will tell you. I don't know why it doesn't do it. Okay. The researchers actually listened into individual nerve cells firing in the anesthetized cat. So the cat is not feeling any pain. Cat is with anesthesia. They presented the cat with different visual images. So David Hubel and Torsten Wiesel started in the 1950s, started working with cats. And that was in the 1950s. The first experiments didn't go too well. In the beginning, they could not make the cells fire at all. You can see the cat right there in the middle. They tried lights all over the screen and nothing seemed to work. And rather by accident, one day there were shining spots, either the light one or dark ones, to the cat's eyes, to the cat's retina. And they found that the black dot, you saw the black dot moving up and down, was actually working. That is, it was creating the signal. Unfortunately, if you cannot hear what's going on in the video, you cannot hear the firing of the neurons. As this dot moved down, the firing increased. And it turns out that this line in the middle is the one moving up and down. That was what the cat responded to. That's the single electrode recording. All right, we have just couple of minutes to finish. Close this. Go back to, stop. 1 second. Thank you, Parker. And I realized that it wasn't possible for you to hear. Let's just finish with the okay. Right. Roster this. Okay, can you see, guys, the picture of Hubel and Wiesel that received their Nobel Prize for finding how neurons in cat's brain responded? Okay, good. Finally, and this is our last thing, oops. There are a bunch of neuroimaging methods that I'm not showing you because, again, it takes time for me to switch back and forth. But there are a couple of them. One of them is sort of based on electricity. Remember, our brain creates activities that we can record. We can either record it in the EEG, which is just types of waves, alpha, beta, gamma, delta, and so on, or it could be event-related brain potentials, which is a little different. It's based on EEG, but it's multiple EEGs collapsed, averaged. So when you present people with the same stimulus several times, you can collapse and average their responses. You can see here a participant in the study wearing the EEG ERP cap with electrodes. Again, it's absolutely innocuous because it records your own activity in the brain. And let's see, and I think that's it. Give me 1 second. Right, and finally you have things like PET scan, MRI, and fMRI. Again, the easiest way to understand what they do is to actually Google them and have a look at YouTube of what that looks like. Okay, at this point, guys, I'm done. And if you have any questions between now and Thursday, let me know. Otherwise, I'll see you on Thursday at 10:10, and the class is over. Take care, Raphael.
And thank you.
Thank you, Leroy. See you Thursday. Bye, Karen. Bye, Isaac. Rebecca. Take care, Yisrin. Bye, Svetlana.
Professor?
Yes, Justin.
So,
Can't hear you. Are you saying something?
yeah, I was unmuted for a second. I was asking about the person I'm working with. I have to have a partner by Thursday, right?
No, on Thursday.
On Thursday.
Yeah. Yeah. So what we will do, because today, remember, is optional. Not everybody is required to be here. So on Thursday, we will start with finishing up group assignments. So what I suggest you do is go to Blackboard, go to the blogs, and go to self-introduction.
Yeah, I looked
And
through
listen.
it. I looked through it.
Did you find somebody you would be
Oh,
interested in?
I have a few names in mind. I didn't pick one yet, but you know, because.
Okay. All right. So what you could do, you could say, I'm interested in pairing up with such and such and such, and you will quickly decide which of the people that you're interested in will join you, because it only is just two of you, you and another person.
Okay, so we're gonna have the same partner for the whole semester, doing the experiment,
Normally, right? Normally, yes, but sometimes things don't work out. Sometimes people drop. It happens. So if you find yourself without a partner after the first experiment, and somebody else is without a partner, then you'll create a new group.
Got it.
Okay, Justin.
Okay, see you on Thursday.
See you on Thursday. Take care.
Okay.